Well, welcome to the Department of Psychiatry Grand Rounds. As you know, Grand Rounds is sponsored by the University of Arizona College of Medicine and the Arizona Health Sciences Center. And this is part of our psychiatry department weekly Grand Rounds. Today we have the honor of having with us Dr. Amelia Galitano, who comes to us from the Department of Basic Medical Sciences at the University of Arizona College of Medicine in Phoenix. She's also an assistant professor of neuroscience with Arizona State University, an assistant professor of neuroscience with, of the Department of Basic Medical Science with the College of Medicine, which is part of the joint effort of the University of Arizona and ASU. Her background is she's an MD PhD with a BA in behavioral science from the University of Chicago. And then she went to Penn, where she had an MD and a PhD, and did psychiatry residency at Columbia. And her work has been, of course, in the field of neuroscience, and today she'll be talking to us about the candidate gene Egr3 mediating stress reactivity and the role in schizophrenia, the response to clozapine in mice. So again, let's welcome Dr. Galitano. Thank you.
Thank you, Francisco. So I want to say that I'm very pleased to be here. Thanks for inviting me, and also very pleased that Dr. Mishashek recently accepted my request to become a faculty member in the Department of Psychiatry here. So I am now a member of your department as well. Thank you. So it's nice to finally meet some of the members of this department. So today I'm going to talk to you about the research that I've been doing for the past 6 or 7 years on a gene that I have hypothesized may be playing a role in psychiatric illnesses, and in particular in schizophrenia. So I don't have to tell you, because you all know what an exciting time this is to be in psychiatry, clinical as well as research, because we've really seen a huge transformation in our understanding of psychiatric illnesses in the last several decades, and are finally at a point where we can start to study these diseases at the molecular level. So what got us there? Well, one of the first studies, one of the first findings really to lead to this transformation where we could start to study and do research on psychiatric illness was the finding that there is a genetic component to psychiatric illnesses. So this sort of was transformational in getting us out of the phase of blaming the mother for schizophrenia and that sort of unpleasant history that we have in psychiatry. When work by Gottesman and colleagues demonstrated that the risk of having the disease schizophrenia was related to how many family members had psychiatric illnesses, or schizophrenia in particular. So since that time, we've seen initially the identification of genetic loci based on large-scale family studies and pedigrees, which identified regions of the genome where we thought there could be a gene involved in the illness. And it wasn't really until 2002, so very recently, that we had our first candidate gene for a major psychiatric illness, and that was schizophrenia. The gene was neuregulin-1, and it continues to be a leading contender for genes that could be involved in influencing risk for this illness. But remarkably, we still have no definitive gene for schizophrenia or any psychiatric illness. So by 2005, that one gene was transformed into, now I have to see if I can go back, a list of 12 genes that were candidates for playing a role in schizophrenia. And now, just almost 4 years later, right, we're now 2009, we have numerous genes implicated in the illness. But as I said, none is definitively been shown to cause the disease. Now, this is different than other illnesses, other medical illnesses, where we have known genes that cause either familial diseases, hereditary diseases in neurology and in cancer, and definitive diseases such as cystic fibrosis, where we know it, one particular gene, if you mutate it, you have the disease. If you lose both copies, if you have two normal copies, you don't have the disease. Why is psychiatry so far behind? Well, there's a number of reasons why. One of them is that we don't have any definitive tests for our illnesses. There's no biological result that we can pull out and say, you know, a piece of tissue that we can take from our patients and demonstrate that they have this illness. Instead, we have a constellation of clinical signs and symptoms that we put together under the definition of an illness. So it wasn't until relatively recently with the third edition of the DSM-3, where we have finally come to some consensus about how to define these illnesses. So that was a major point in allowing us to do research on these illnesses. But there still remains, as you know, with each new edition, the definitions change. And it's pretty much accepted among clinicians that even something as precisely defined as we've been able to, something like schizophrenia, a disorder like schizophrenia, probably encompasses multiple disorders that have overlap in their presentation. So that's one of the reasons. Another one of the reasons is this. If we go back to this figure, monozygotic twins here share 100% of their DNA, but they only share, if your monozygotic twin has schizophrenia, you really only have about a 50% chance of having it. So what does that say? That says that the other 50% is caused by something else. So this is an image from, this is the cover of a book, a memoir written by two sisters, very recently published in the last 5 years, Pam Spiro Wagner and Carolyn Spiro. One of them had schizophrenia and the other one became a psychiatrist. So what accounts for the other 50% that isn't due to genetics, as we have conventionally thought of genetics? Well, one thing that people commonly think of is environment. And what do we mean when we say environment? We could mean lots of different things: exposure to toxins, things that we ingest, as well as events in the environment like stress. And so there's actually been a fair amount of research done to demonstrate that some stressful events, such as exposure to in utero infection, traumatic birth events, or stressful life events, can in fact influence the risk to develop disorders, psychiatric disorders, such as schizophrenia, depression, anxiety, and PTSD. So we know somewhat about this. We know also something about the neurobiologic or functional MRI effects of these disorders in the brain. What we know almost nothing about is in this black box here. What links the two? What are the mechanisms by which environmental stress can cause these neurobiological changes in the brain that influence the risk to develop psychiatric illness? Well, again, another landmark that's finding in the last decade, and actually the last 5 years, was this study by Caspi and colleagues published in Science in 2003. And this is a figure from that paper, which demonstrates, really for the first time, the interaction between environmental stress and a morphological difference in the DNA, a genetic difference, to influence the risk for a psychiatric illness. So this figure shows, in this case, this is looking at the presence of a polymorphism in the serotonin transporter molecule. Now that's the molecule at which the SSRIs act, bind and block. And you can have, there are two forms of this promoter region, a short allele and a long allele, indicated by L or S. And what they found was that in the, when they looked just at genotype alone, there was no influence of the particular combination of alleles that an individual had on their risk to develop a major depressive episode or other measures that they looked at, such as suicidality, until they took into account the stress history of the individual. So in the absence of maltreatment, again, no difference genotypically. However, in patients that had a history of severe maltreatment, patients with 2 long forms of the promoter had no difference, no increased risk to develop a major depressive episode. However, having presence of 1 or 2 copies of the short form significantly increased your risk to develop depressive disorder. So what they said was there is a functional change in the DNA that interacts with an environment of stress to influence the risk for psychiatric illness. So that's the model that I look to in the work that I've done. And what I've decided to do in my research was to target specifically that black box, to ask what is it that's going on in the middle here that could translate events in the environment into changes in the brain that could have long-lasting effects. And what's in that box is the expression of a class of genes called immediate early genes. So what I'm going to tell you about today in my talk is, first I'm going to talk about whether immediate early genes are in fact playing an essential role in this response. So what we know is this class of genes is activated, I'm going to tell you about that a little bit more in a minute, by stress. But what we hadn't known until the work that I'm going to present today is whether or not that activation was playing an essential role in the brain or individual's behavioral response to that stress, or was it some nonspecific byproduct of stress that had no functional result. So first I'm going to tell you about studies that I did to determine whether or not these immediate early genes were in fact playing an essential role in the animal's response to stress. And then I'm going to talk about whether or not a member of this family of immediate early genes in particular, Egr3, could be playing a role in schizophrenia. And then I'm going to tell you a little bit about work that I've done to test whether Egr3 is a candidate gene for schizophrenia. And that's studies that I've done in the mice, as well as ongoing studies that I have in humans, looking at genetic polymorphisms in the Egr3 gene. And finally, I'll end with telling you a little bit about the work that I'm doing currently in the lab, which is using some of the observations that I'll show you that we've found, some of our discoveries, to try and decipher the neurobiological abnormalities in the brains of Egr3 mutant mice, with the hope that they'll be an insight into some of the neurobiological abnormalities in patients with schizophrenia. Okay, so immediate early genes are activated in response to stress. This is throughout the body, but I'm particularly interested in their activation in the brain. So what do I mean by that? I mean that stress in the environment leads to, and this is a cartoon of an immediate early gene with the open reading frame of the gene, the coding region in blue, and an intron. So this is our molecular biology shorthand for turning on gene expression. So what does that mean? Well, stress in the environment leads to increased expression of this gene, so production of mRNA, which is then translated into protein. And in the case of the EGR family of proteins that I study, these proteins act as transcription factors, which means they bind to DNA and they regulate the expression of other genes. So what this means is, and what does this look like? This is an example of what it looks like. This is a coronal section. This is a figure taken from a paper, not my work, but somebody else's, showing a coronal section through a rodent brain at baseline, showing expression of the immediate early gene, the first member of the family that I study to be identified, EGR1, at baseline. And you can see that there's very little expression. However, 45 minutes after a stressor or an environmental impact, in this case it was exposure to the drug cocaine, we see massive increase in expression. This is mRNA in the brain. So in 45 minutes, this has happened. Now, because of the fact that the EGRs then act as transcription factors and turn on a whole other set of genes, you can see how, if I go back, not easily, how a single event in the environment can lead to this initial gene expression change, which then initiates another cascade of multiple genes being expressed, and this can have long-term effects in the brain. So it's a mechanism by which an event in the environment can have long-term changes in the brain. So what are the early growth response genes? They are a family of genes that were originally identified by the family member Egr1, which was identified in multiple labs at the same time, and so given multiple different names. And I did my postdoctoral research in the lab of Jeff Milbrandt, and he identified Egr1 in an assay where he applied nerve growth factor to PC12 cells in cultures, which causes them to change into a neuronal phenotype. And he identified that this is one of the genes that was activated in response to nerve growth factor. So he named it nerve growth factor inducible A. Well, it was subsequently discovered by other people and called ZIF268, CROX24, and a number of other names. But in general now, most people have accepted the convention of EGR. So EGR1 has been studied very much is used more as a tool in the neuroscience field to look at gene expression induced by many different kinds of stimuli, sort of in the same way that we use functional MRI in humans. If I do this to my mouse, then look at EGR1 expression, that means that the part of the brain that's lit up with EGR1 expression was involved in the brain's response to the stimulus, okay? But what nobody had done was seen whether EGR1 or any of the other family members, each of which were cloned by homology, was actually playing an essential role in that response to the stimulus. So the gene that I'm going to talk about today is EGR3. Again, these genes are highly homologous and all encode zinc finger transcription factors. So how did I decide on EGR3? Well, when I got this idea that maybe these genes could be playing a role in psychiatric illness, because psychiatric illness has both a genetic and an environmental component, and these genes translate events in the environment into long-term changes in the brain, I asked the women in our lab that handled the mice, the ones that bred our mice, and we had knockout mice for each of these different genes in the lab. I asked them, had they ever noticed anything strange about any of the mice, the EGR 1, 2, 3, 4 lines? And they said, oh, EGR3, those mice are crazy. So you can imagine that that was music to my ears. So what did they mean by that? What they meant was every time they went to change the cage on the EGR3 knockout mice, the mice went nuts. They ran around the cage. They tried to avoid being picked up. They were much more likely to jump out of your hands or bite you than all the rest of the mice, this large colony. So we decided to assess that reactivity to handling. And we did this by having a group of 3 blinded observers, these are experienced mouse handlers, observe the mice and rate them on a scale of 1 to 5, where 3 is a normally reactive mouse. 2 or 1 would be hyporeactive, not react much when you pick them up or not at all. A 4 would be a mouse that might be difficult to catch. Once you get it out of the cage, would be jumping around while you're holding on to it. A 5 would jump around continuously, try to escape, and may even bite you. So what we found was we tested on 3 subsequent days, and if you look at day 1, we found that EGR3 knockout mice scored much, much more reactive than did their wild-type litter mates. And the litter mates scored right around 3, which suggested that our assay was pretty much on target, that our observers were rating the mice as was expected. This hyperreactivity to handling did diminish somewhat over the subsequent 2 days of testing, but remained statistically significantly greater than the wild-type litter mates on all days. So what this meant was that these mice were hyperreactive to a very mild stress, just being picked up. So we wanted to see whether that stress reactivity was also reflected in a physiologic measure. So we measured cortisol levels, or corticosterone in mice. Mice don't have cortisol, but they have the equivalent of corticosterone. So what we did, if you look at baseline, we found that testing corticosterone first thing in the morning in these mice, they did not differ, the knockout animals, from their control litter mates in the level of corticosterone that they produced. At a subsequent time, I handled the mice 30 minutes before we tested their corticosterone. And in wild-type mice in black, what you see is that handling leads to a significant increase in corticosterone release, over doubling the corticosterone. That's just from picking it up for a period of 90 seconds, putting it back in the cage, and then 30 minutes later testing corticosterone. What we saw in the Egr3 knockouts, however, was a huge increase. These are completely non-overlapping groups. Hugely statistically significant increase in the amount of corticosterone that they released in response to this mild form of stress. So that indicated that the mice were not only behaviorally more reactive to stress, but this was also reflected in a physiologic measure of stress. So we tested the mice in a series of other stressful paradigms to see what their reaction was. And placing an animal in a novel environment is a mild form of stress as well. And what we found was that the Egr3 knockouts in white were much more active, hyperactive, when placed in a novel environment than were the littermate controls. And that this, they habituated over time such that by the end of a 1-hour period, they were roughly of similar activity, not statistically different than their control littermates. We used another test in which we tested how long it took for the animals to find food when it was hidden under the bedding of the cage. And when I initially did this test, it was to determine whether they had normal olfactory functioning, and this was as a control for some social behavior abnormalities that I discovered in the mice. What I found was this huge difference where the knockouts took much longer to find the food than did the wild-type animals. However, sometimes they found the food and they didn't eat it, and that struck me as odd. They'd uncover it and wouldn't even eat it. So then I thought, well, there's two factors that are different here. There's the genotype, but there's also that I'm placing them in a new cage when I do the test. What happens if we test them in their home cage? The difference goes away. So that indicated that their reactivity was to the novelty of the environment. It wasn't due to their inability to detect and find the food. Then we tested the mice in another paradigm in which we looked at their reaction to a loud noise and tested their startle response. And in this test, animals are exposed to 20 episodes of a loud noise, which is given randomly in 2-minute blocks. And an average of 5 of these reactions are averaged together and graphed over time. And what we see is that wild-type mice show a large reaction to the startling sound, and this is measured by the amount of pressure that they exert on the base of the apparatus, which has a detection module in it. So they start out with a large reaction, but after they hear this sound over and over again, they habituate. They know, gee, I heard that sound before and nothing happened, probably nothing's going to happen this time, so I'm not going to jump so much. In contrast, the Egr3 knockouts failed to habituate and reacted just as much after they'd heard the sound 20 times as they did after the first, indicating this, again, persistent response to this startling sound. So to summarize what I've told you about up till here, these tests showed us that Egr3, when it's absent in mice, it does affect their stress-related behavior, suggesting the possibility that the normal activation in normal mice that we see of the Egr3 gene in response to stress is having a functional consequence. In the absence of the gene, the reaction to stress is heightened. So a number of these tests are also tests that are used for schizophrenia-like behavior in mice to assess their schizophrenia-like phenotype. So that's true of heightened stress response is a characteristic that we see in patients with schizophrenia. They may get exacerbations of their illness in the context of stress. It's one of the first things we ask our patients when they come to the emergency room with a new episode of psychosis. What was going on at home? Was there any stressor? This hyperactivity in a novel environment is a standard test that we use for animals based on studies in which drugs that increase dopamine release cause hyperactivity in animals, and when this can be blocked by antipsychotics. And there's a literature indicating that patients with schizophrenia fail to habituate to startle response. So that supported the idea of the possibility that EGR3 could be playing a role in schizophrenia, since when it's knocked out in the mice, they show schizophrenia-like behavioral phenotype. So, as you know, schizophrenia is characterized by 3 major categories of symptoms. Now, the DSM-4 diagnosis really only emphasizes the first 2, the positive symptoms and negative symptoms. However, there's this 3rd category that we talk about a lot and is now an increasing area receiving tremendous focus from the research perspective, and that's that schizophrenia patients have cognitive deficits. So some of the behavioral abnormalities that we can test in mice obviously don't have schizophrenia, right? Because it's a human disorder. But we use these models, like I've said, to the tests that I've described, based on a history of the responsiveness of these abnormal behaviors to antipsychotic medications, we can use these tests to establish whether or not a gene could possibly be playing a role in schizophrenia. What else can we do? Well, we can look at whether, for example, an antipsychotic medication might reverse that abnormality. So what's the likelihood, going back a step, what's the likelihood that the EGR3 gene could be playing a role in schizophrenia? In other words, that mutations in this gene in humans could predispose to schizophrenia. Well, when I got this idea, I looked at the literature, and sure enough, there is a biological argument that EGR3 could be playing a role. Number 1, EGR3 is directly regulated by the gene neuregulin 1. So it turns out that neuregulin 1 in at least 2 different parts of the body, one, the developing muscle spindle, that neuregulin 1 produced in the primary afferent A1 neuron is required to maintain expression of EGR3 in the postsynaptic cell. In addition, after I developed this hypothesis and wrote a whole grant and submitted it, a month later, a paper came out indicating that the protein calcineurin may be a schizophrenia candidate protein, and that mice lacking the calcineurin protein show schizophrenia-like behaviors. Well, it turns out calcineurin also in the literature had been shown to regulate EGR3. So, in addition, some of the early papers on EGR3 expression had demonstrated that its induction in the hippocampus in animal models in response to electric stimulation in electrophysiology studies its activation was dependent on the function of NMDA receptors, suggesting that EGR3 is also downstream of NMDA receptors. And NMDA receptors are a major factor, we believe, in schizophrenia. So there's the NMDA receptor, a type of glutamate receptor, NMDA receptor hypofunction hypothesis of schizophrenia. So that placed EGR3 downstream of 1, 2, 3 schizophrenia candidate molecules, which in my mind was pretty good evidence that it could possibly be playing a role. And again, there's the possibility that the effect, the dependence of EGR3 on NMDA receptors could be via calcineurin, because NMDA receptors, when activated, lead to influx of calcium into the cell, which stimulates the protein calcineurin, which then regulates EGR3. So this is some image from a paper, review article by Sheng and Kim, showing some of this postsynaptic signal transduction pathway that is involved in calcium influx in response to NMDA receptors as well as other types of glutamate receptors. So again, as I said, NMDA receptors, when activated, lead to influx of calcium. They activate calcineurin. All of this, these genes in signal transduction lead to the activation of RAS, RAF, MEK, ERK pathway of signal transduction molecules, and these lead to nuclear gene expression. And what is in this pathway? EGR3. We know that this is the pathway that leads to its activation in the neuron. So finally, if you think your gene of interest may be involved in schizophrenia, it would be nice if it mapped to one of those loci that I told you had been discovered by years and years of linkage studies in human pedigrees. And sure enough, Egr3 maps to locus 8p21.3, which is a major schizophrenia candidate locus. So, what else about the animals suggested the possibility that they may be playing a role in schizophrenia? Well, another thing that we noticed is that the mice were highly aggressive. Now, schizophrenia patients aren't necessarily more aggressive. However, there is an association between poorly controlled schizophrenia and aggression. And the aggression tends to abate when we adequately treat the symptoms, suggesting the possibility that the aggression in large part is due to the psychosis, right? If you are paranoid and believe somebody's going to kill you, you might have an aggressive response. Some might consider that logical. So what I want to show you is the aggressive behavior of our animals. So this is a test called the resident and intruder model. This mouse is an Egr3 knockout mouse. And over here, the mouse without the marking on its tail is its wild-type litter mate. So these two mice were born litter mates, but differ in this one has 2 normal copies of the gene, this one has no normal copies of the gene, no functional copies. And they were raised together in a litter. In this assay, male mice are housed alone, and then they tend to develop a sense of territory. When you introduce another foreign male, an intruder, they may react with aggression. And what we see is that the Egr3 knockout mouse is much more aggressive than the wild-type littermate, who sniffs the new guy and then sort of ignores him and goes about his business. So, how does this look when we represent it graphically? Well, over here, you can see that we ran this test on 3 different days. And if you look at run 1, the first day, it shows this is the duration in a 10-minute test, the total duration of time that the animals are attacking, either biting or fighting with the intruder animal. And what we see is that the Egr3 knockout spends a significantly greater amount of the time in aggressively attacking the intruder mouse than does the wild-type littermate. And this persists over several days of testing, which is expected. In contrast, the latency to the first attack is much shorter in the Egr3 knockout mice than in the wild-type mice. And latency to attack has been associated with impulsivity, another characteristic of some psychotic disorders. Okay, so what do we have so far? We have that Egr3 knockout mice show schizophrenia-like behavior in a number of different models of schizophrenia. They are hyperactive in a novel environment. They show this startle response that fails to habituate. They're hypersensitive to stress. I didn't tell you about, but they also have some abnormalities in social behavior in addition to the aggressive behavior. And so the next question is, does any of this respond to treatment with antipsychotics? So we tested that next. What we found was when we chronically administered the antipsychotic drug clozapine, that we could reverse the aggressive behavior of the Egr3 knockout mice. So what we did was we administered clozapine at 7 milligrams per kilogram per day to mice over a period of 2 weeks, and then waited 24 hours after the last dose and tested them in the same aggression test that I just showed you. And we found that mice treated with vehicle showed the same response that we showed in the previous graphs, that the Egr3 knockout mice were much more aggressive than their wild-type littermates. However, the Egr3 knockout mice that were treated with clozapine for 2 weeks showed significantly less aggressive behavior, and the wild-type showed none. So what this demonstrated was that we could reverse this behavioral abnormality with an antipsychotic, clozapine. So you might ask, why did we bother exposing them to clozapine for 2 weeks? And well, we found that we couldn't treat them acutely with clozapine and do the test because it was so sedating. So we gave 3.5 milligrams even, or 7 milligrams per kilogram, which is roughly equivalent to a human effective dose in humans. And the wild-type mice were just knocked out flat. So I'm going to show you a video of that. Interestingly, we didn't see the same effect in the Egr3 knockouts. So what you're going to be looking at here is 3 Egr3 knockout mice, and their tails are marked, and you don't have to find that, I'll point them out. It's our way of then later going back when we score the video and telling which mouse was which. And 1 wild-type mouse. So the wild-type mouse and 2 of the knockout mice received 7 milligrams per kilogram of clozapine, and 1 of the knockout mice received vehicle. And this is 2 hours after administration of the drug. And what we see is this is the wild-type mouse that received clozapine. And 2 of these knockout mice received the same dose of clozapine, and 1 of them received vehicle. And I would be hard-pressed to pick out without knowing which one got the vehicle. This is 3 wild-type mice. 2 of them got vehicle and 1 got clozapine. And this is just to show you that, you know, the wild-type mice, what they would normally be behaving as in comparison to their knockout. And that's just to show that he's not dead. So the difference was quite profound. So to represent this graphically, what we see is, again, now this is a different way of demonstrating the same activity in a novel environment graph that I showed you before. Rather than showing data points 5 minutes averaged over time, we average them all together in one block. And the mice that received clozapine, this was actually a 30-minute test rather than a 1-hour test, show significantly more activity than do the wild-type littermates. These are the ones that received, I'm sorry, did I say clozapine? That received vehicle, right? So they're showing, again, that baseline hyperactivity of the Egr3 knockout mice. Then this graph shows actually half the dose that I showed you of clozapine in the video, 3.5 milligrams per kilogram, and that dose is enough to almost completely suppress the activity of wild-type mice and doesn't touch the Egr3 knockout mice. No statistical difference between these 2 values. So the statistical difference here refers to the wild-type mice. So when I saw this result, the first thing that I thought was, wow, that's like what we see in humans. And so at the time, I had a listserv that I was a member of with my residency cohort from Columbia, and the faculty there all participate on a psychiatry listserv. And so I emailed quickly and I said, what is other people's clinical impression of patients' response to clozapine? Do they find that patients are less sensitive to sedating effects of clozapine than are, or antipsychotics in general, than are normal people? And I got back lots of responses. Some people said, I don't know. The majority of people said, absolutely, I agree. Nobody said they disagreed. But I got a couple of interesting anecdotes. One was, one person said, well, I remember when I was a resident, this guy came into the ER, this psychotic patient, and over the course of a day, we gave him 100 milligrams of haloperidol, and he was still walking around. Then somebody else wrote back and said, I remember when I was a resident, there was this nurse on the inpatient unit, and she wanted to know what it felt like to be on an antipsychotic. So she took 1 milligram of haloperidol on a Friday, and she woke up on a Sunday. So that shows you this 100-fold difference in the sensitivity to side effects of these medications. And when I went to the literature, I was able to find an article by Neil Cutler in which he emphasized this difference, that schizophrenia patients have a much higher degree of tolerance to the side effects of antipsychotics, both haloperidol and clozapine, than do healthy volunteers. And actually, the main point of this article was that we shouldn't even be using healthy volunteers in studies of antipsychotic medications, because it's just too dangerous. So, to summarize to here, again, schizophrenia-like behavior in the mice, and now we've shown that some of this abnormal behavior we can reverse with an antipsychotic medication. What about the cognitive deficits? Remember, cognitive deficits in schizophrenia. Very important area of research now. Well, we decided to test whether our mice had memory deficits. And in fact, I was really pretty convinced that they would. based on a couple of findings. Based on these observations of social behavior abnormalities that I had seen, in addition to the ones I've shown you, that if mice can't remember their cage mates, they would be more likely to reinvestigate them, to re-identify them. And the initial observation I made the first day I walked into the animal colony, after the ladies that took care of them told me that the mice were weird, I opened the cage and looked, and the Egr3 knockout mice sniff their cage mates repeatedly, over and over and over again, much more so than the wild types. And I had then videotaped them and calculated, measured this activity, and yes, indeed, it's highly statistically significant. Why do they do that? One explanation could be if they can't remember their cage mate, then each time the animal walks by them, they have to say, "Who's that?" And the way that animals identify each other is via olfaction, by smelling them, just like dogs at the park. So that's a possible explanation. So we wanted to test whether there were memory abnormalities in the mice. And an easy way to do that is using the simple maze, the Y maze. And the way this test works, and it's a spatial navigation memory and a very short form of memory. You put the animal in one arm of the maze, and you measure entries into the other arms, and you count for a total of, I think it was 12 entries. I'll see you on the next slide. So a smart mouse would investigate a new environment in a systematic manner. They'd go from one arm to the next to the next. So the mouse gets 1 point, and these points are cumulative, each time it investigates the 3 arms in sequence. If it goes out of sequence into this arm and back into the arm from which it came, that's less efficient, and they don't get a point for that. So the total number of points that they can get is 12. I guess we measure them, we start on the third one. And they're cumulative. So a mouse that scored perfectly would be scoring in parallel to this line, okay? And what we see is that Egr3, wild-type mice perform pretty well, and Egr3 knockouts perform statistically significantly less well than their litter mates. And this test has been associated with memory deficits. So mice that are given amnestic drugs perform poorly in this test, and mice with other kinds of cognitive deficits perform poorly in this test in a similar manner. So that suggested that the mice do have memory problems, which could also account for the failure to habituate to a novel environment as well as to the startle stimulus. So then we were curious as to whether the Egr3 knockout mice may have some deficits in synaptic plasticity, because synaptic plasticity, particularly hippocampal, is thought to be the basis of memory formation. So the other reason that I had a strong suspicion that the mice would have memory deficits and synaptic plasticity deficits was that Egr1 knockout mice had been shown to have deficits in the form of synaptic plasticity long-term potentiation, as well as deficits in long-term memory, and the two were associated. So I figured Egr3 and Egr1, highly homologous. We know that they're both induced by many of the same stimuli. We know that they're both activated in the same cells in the brain, and both in an NMDA receptor-dependent manner. So I thought it was highly probable that we'd see a synaptic plasticity deficit. So what we're looking at here is, this is a nice figure that was made by my colleague Yuki Izumi at WashU, who did the electrophysiology studies as a collaboration for me. And this just shows how, this is a picture of the mouse brain, how we dissect out the hippocampus, comes out in one nice, neat piece. It gets sectioned, and these sections get floated in a bath, and in which you can do electrophysiology studies for many hours. The circuitry that we study here in the hippocampus is the Schaefer collateral system. So CA3 neurons, which have their cell bodies here, project their axons up to the dendrites. They synapse here at 4 onto the dendrites of CA1 region cells. And when you stimulate these axons, these projections here at 3, and record the result from the dendrites in this region, you can see electrophysiologic changes, these excitatory postsynaptic potentials. So what you can measure is this phenomenon where induction of either a high frequency or a low frequency stimulus to the axons has a long-term effect on the synaptic activity of the postsynaptic neurons. Okay? So this is a phenomenon for which the Nobel Prize was awarded several years ago. That is, this induction of a long-term change is plasticity. And because it happens across the synapse, that's why we call it synaptic plasticity. So it turns out that the EGR3 knockout animals, or the wild-type animals in white, we can induce, so this is baseline firing of the neurons, the postsynaptic neurons. After a high-frequency stimulus, we see an induction or an increased frequency of firing that persists for a long period of time. And it was not changed in the EGR3 knockout animals. And looks like we don't have a good definition of which is which here. I apologize for that. But they're not different. So we didn't see a difference in at least this short or acute phase of long-term potentiation in the hippocampus. However, when we looked at another form of synaptic plasticity, long-term depression, we did see a difference. So here, to induce long-term depression, which is a decrease in the frequency of firing in response to a low-frequency stimulation of cells, we see that the wild-type animals, you administer the low-frequency stimulation, see this decrease that persists over time. We measured out to 90 minutes. The EGR3 knockouts, however, show the initial decrease during the low-frequency stimulation, but rapidly the firing rate comes back up to baseline. So they fail to form this long-term change across the synapse in response to the stimulus. Interestingly, this is the same phenomenon that had been reported for the calcineurin knockout mice. And this was in a paper that preceded by several years from the same group that reported the schizophrenia-like phenotype in their mice. It was reported several years earlier in Cell. Again, they show that the calcineurin knockout mice in black, after induction of this low-frequency stimulus, their firing rate comes back up toward baseline. And that's in contrast with the wild types. So we went on to further try to decipher the abnormality responsible for this, and we surmised that it was due to the dysfunction of NMDA receptors. So using an agent, efenpridil, that's an antagonist specifically on NMDA receptors that contain an NR2B subunit. We assessed the firing rate, this is basal firing, of the neurons in this hippocampal slice in response to application of efenpridil. And in wild-type animals, what you see is at the time, so this is baseline firing, at the time of application of efenpridil, it decreases the spontaneous firing of wild-type neurons, and the EGR3 knockouts are not susceptible to this. So this demonstrated that the EGR3 knockout mice have a dysfunction in NMDA receptors that contain an NMDA subunit. Now, the most parsimonious explanation for this would be that EGR3, which is a transcription factor, is required to regulate expression of the NR2B gene. But when I tested this using quantitative RT-PCR, we did not see a difference, unfortunately. So that suggests that there may be another mechanism underlying this effect. But it's not simply due to EGR3 regulating this gene. So when I went back to the literature with these results, I found that there is a small literature, but significant literature, demonstrating that stress affects not only memory, both stress and exposure to novelty affect memory, but they also affect specifically the form of synaptic plasticity, long-term depression. So stress and exposure to novelty both facilitate, they inhibit LTP, but they facilitate LTD. So this was an interesting correlation, that what we see is now a defect in the response of our animals to stress and exposure to novelty, and a defect in LTD. So we're curious to try and decipher what's going on with this. But it is our first evidence of concrete abnormality in the brains of the animals that may and is in a system that is, we know, implicated in schizophrenia. That is the NMDA receptor system. So this is a figure taken from another paper showing that calcineurin A protein, so again, calcineurin being another candidate molecule for schizophrenia, is deficient in patients with schizophrenia. And in this study, they were actually looking at comparing schizophrenia patients that smoked with those that don't smoke. But interestingly, so you can, regardless of whether or not the patients are smoking, what it shows is that calcineurin A protein is not different in control individuals based on whether or not they smoke. It's low in schizophrenia patients that don't smoke, and it's brought up again in patients that smoke, suggesting the intriguing possibility that the reason that schizophrenia patients smoke is because it augments expression of these molecules that are deficient in them and can somehow, there's a long time been thinking clinically that patients gain some benefit by smoking, and this may be the molecular explanation for that. So interestingly, when I looked at the other genes that were regulated, this is again in hippocampal tissue from schizophrenia patients, it turns out that EGR3 was in their list of genes that were misregulated in schizophrenia, and it follows the exact same pattern as calcineurin A. So that was very exciting, again, because EGR3 is regulated by calcineurin, and it was the first definitive demonstration that EGR3 gene expression is in fact reduced in patients with schizophrenia. So I was actually in contact with the authors, with the lead author on this paper. She's the one who sent it to me, and she hadn't realized that my gene was one of the positives in their study. So I called her back. They went back and confirmed, this was from microarray study, they went back and confirmed with a quantitative RT-PCR and found, in fact, that there is an overall difference even if you don't control for the smoking. So EGR3 is a gene that is shown to be misexpressed in the brains of schizophrenia patients. And that's here on their list. Subsequently, a year ago, another study came out, again by the same people that the Tanagawa lab at MIT, in collaboration with researchers in Japan, and they had now gone on to study EGR3 and found that EGR3 levels in another brain region were decreased. In the prefrontal cortex of patients with schizophrenia were decreased compared to controls. They also looked at bipolars, but that difference was not significant. So a second study showing decreased expression of EGR3 in the brains of schizophrenia patients. In that study, they also did the genetic association and found that indeed polymorphisms in the EGR3 gene were associated with schizophrenia in this Japanese population. So that was both exciting and frustrating because I was doing the same study. They scooped me on it, but I have a different population that we're doing it in, so we hope to publish our findings soon. And that's a human study that I didn't present today. So what am I doing now? So currently, the studies that we're pursuing in my lab up in Phoenix are to finalize this genetic association study that we've done. We resequenced the EGR3 gene in its entirety from 96 schizophrenia patient DNA samples. We identified 9 polymorphisms in the gene, 3 of which had been known, and 12 polymorphisms, 3 of which had been known and 9 of which were novel. And then we collected a large sample, actually we're using NIMH Schizophrenia Genetics Initiative DNA samples. And we developed a probe that would recognize these different polymorphisms. And we did a large-scale genotyping of the large group of patients and controls as well as families. So we can do 2 different forms of statistics, the EDTT, which controls for population stratification, as well as a case control analysis. And we should have those results very soon. In addition, that's the first 2 current directions. I have a grant to try to decipher the neurobiological abnormalities in the EGR3 knockout mouse using 2 different approaches. The first is a pharmacologic approach. So and I'll tell you the second very rapidly, which is that we've done a microarray study using the hippocampi of EGR3 knockout mice at baseline and then following high level of induction of the genes to find target genes and compare those. That's one branch of the study. And the pharmacologic approach is that we're going to use this assay for the failure to be sedated by clozapine. So the thinking behind this is that clozapine binds to a wide array of receptors. Presumably, one or more of these receptors is functioning abnormally in the EGR3 knockout mice that's allowing them to not be sedated by the clozapine. So what our approach is to use different agents that act on subsets of these receptors and to identify which ones showed differential sedation in the EGR3 knockouts, and then those would be candidates for being abnormally expressed or dysfunctional in the EGR3 knockouts. Then we'll use agents with further, more and more specific. So for example, there's 5 different types of muscarinic receptors. We start out using a broad-acting anti-muscarinic. If we see the difference in sedation, then we'll try to narrow it down to a specific agent. So where have we gotten with that? Well, very initially, we've looked at conventional antipsychotic haloperidol. And when given acutely and we look at locomotor activity, we see, again, for animals that got vehicle, dose of 0 milligrams per kilogram. Again, the EGR3 knockout mice in gray are hyperactive compared to the wild-type mice. But in contrast to what we see with clozapine, is that a low dose, a dosage of haloperidol, 0.1 milligrams per kilogram, that is not sufficient to cause sedation in the wild-type animals is sufficient to reduce the hyperactivity of the knockouts. Now, this is classically what's been used, this kind of a result has been used to argue that the hyperactivity seen in animal models of schizophrenia is reversible with an antipsychotic. So that's good. But interestingly, it's very different than what we see with clozapine, suggesting that the predominance of activity at D2 receptors is not the difference that we're seeing in the EGR3 knockout mice. And that's compared again with this, where we see very little, this is the clozapine result replicated, very little decrease in activity of the knockouts with a whopping sedation in the wild types. We've also looked at another conventional antipsychotic, chlorpromazine, at the opposite end of the sedation spectrum and potency spectrum. And again, we see the same effect, which is that we're able to reduce the activity of the EGR3 knockout mice in a similar manner as the activity of the wild-type mice. So again, this agent is not showing us the same difference that we saw in clozapine. So that's the approach that we're taking to try and decipher pharmacologically the abnormality in the mice in conjunction with the molecular approach. And we're hoping that this will not only tell us something about EGR3 knockout mouse brains, but hopefully something also about the brains of schizophrenia patients, as well as hopefully deciphering something about the mechanism of action of clozapine, a drug which is one of our leading antipsychotics, but about which we know very little pharmacologically. Finally, there's an intriguing possibility that EGR3 could be distinguishing conventional from second-generation antipsychotics in terms of their response. So in summary, what I've shown you today is that mice that lack this immediate early gene, EGR3, which is a gene normally activated in response to stress, have a heightened response to novelty and to stress. They have behavioral abnormalities that are seen in other mouse models of schizophrenia. They have memory deficits, and they lack a form of synaptic plasticity called long-term depression. And they're sensitive to the therapeutic effects of clozapine, but resistant to its sedating effects. So my hypothesis, and I've developed a model, whereby the hypothesis is that mutations that affect the function of EGR3 in humans could create a susceptibility to develop schizophrenia that is dependent upon the stress history of the individual, going back to the CASP results. So your genetic predisposition is there, but whether or not it manifests as illness depends on whether or not you have stress in your life. The model that we have is that stress does 2 things. It does bad things, We know stress leads to increased cortisol release in humans, and we know that cortisol can be toxic to the brain. However, we also know that stress leads to activation of immediate early genes. And from my studies and other studies, we know that many of the effects of immediate early genes are positive, such as inducing synaptic plasticity, inducing growth factors, vascularization, myelination, and wound healing, all protective effects. So the model is, if you have a defect in the reactivity of your Egr3 or other immediate early genes in response to stress, that in the presence of stress and the absence of that Egr3 positive response, you'll have a preponderance of negative or damaging effects, which over time can result in the neurobiological defects of schizophrenia. In the absence of stress, you're not going to see that difference. So that's it. We'll see how it turns out. So I'd just like to acknowledge the funding that I've received from the Pfizer Fellowship in Biological Psychiatry and from NARSAD, and recently received funding from the IMHR. And the majority of this work was done while I was at the Washington University in St. Louis in the lab of Jeffrey Milbrandt. Dave Wozniak was the head of the animal facility, Animal Behavior Corps, where I did most of my studies. He was generous in allowing me to use his space. Lou Muglia and his graduate student Maureen Boyle helped me with the corticosterone study. And Yuki Izumi and his postdoc Kazu Tokuda did the electrophysiology. And Richard Todd, Bob Cloninger, and Rakesh Nagarajan have all played a role in the human genetics study, which I didn't tell you that much about. Here at the U of A, Scott Janowski was a summer student in the lab who really started the animal response, the animal pharmacology response studies in the lab, and Wendy Ingram and Leela Mupana are current members of the lab pursuing that. Thank you very much.